Hello, my name is Sydney Kobak. I am a senior at the University of Oregon in the Human Physiology Department. I've had the opportunity to work in the Exercise and Environmental Physiology Lab, which conducts human research. With repeated exercise, we will see changes in aerobic capacity that come from the enhanced ability to utilize or deliver oxygen to the working muscle more efficiently. One mechanism to improve oxygen delivery, thus aerobic capacity, is through a reduction in arterial stiffness. Less stiff arteries can supply muscles with oxygen more efficiently, which lead to a greater aerobic capacity. Our lab has shown that histamine is released during exercise and causes physiological changes following exercise, such as reduction in blood pressure and differences in gene expression related to adaptation in previously active skeletal muscle. This was determined through the use of common over-the-counter antihistamines, which blunted these responses. Therefore, the purpose of this project was to determine whether consuming antihistamines prior to exercise would reduce the positive adaptations of exercise training. It was hypothesized that a group consuming antihistamines will demonstrate lower levels of vascular adaptations with exercise training compared to the control group. This project was done in conjunction with Dylan Sieg's dissertation work, which looked at the role of histamine in adaptation to aerobic training. Subjects participated in a 6-week exercise training regimen, as the schematic shows on Table 1. There were a total of 21 exercise sessions and 4 data collection days. The blockade group took antihistamines an hour before every exercise session, while the control group took placebo pills. Each measurement day consisted of a VO2 peak and arterial stiffness measurement. Peak oxygen consumption represented the most amount of oxygen that could be used at max effort. Pulse wave velocity was used to assess arterial stiffness. Image 1 is a diagram that represents how pulse wave velocity was recorded and measured. 2 pressure sensors, called tonometers, were simultaneously placed at 2 different arteries. The digital recording of the tonometers is shown above Part A of the image, which was then used to measure the transient time. The distance between each of the tonometers was measured with a tape measure and divided by the time it took for each pulse to reach the tonometer. We then calculated the velocity of the pulse as distance over time, which indicates arterial stiffness. Multiple arteries were used to assess both central and peripheral arterial stiffness. Figure 1 and 2 demonstrate the increase in aerobic capacity comparing pre to post measurements. Both control and blockade groups recorded a significant increase in oxygen consumption and workload, but did not differ between the groups. Figure 3 shows arterial stiffness of conduit arteries such as the carotid and femoral. There was no change in pulse wave velocity in time or between groups. Figure 4 also shows that there was no change in pulse wave velocity in time or between groups of peripheral arteries. Values are comparable to other studies recording pulse wave velocity of both central and peripheral arterial stiffness. Therefore, we can conclude that arterial stiffness did not play a role in changes in aerobic capacity, and histamine did not impact a change in arterial stiffness with exercise training. Therefore, the hypothesis was rejected. The causes of changes in aerobic capacity did not come from arterial stiffness. Future research should investigate the changes of oxygen utilization occurring in working skeletal muscle. I would like to thank Dylan Sieg. Without him, I would not have been able to complete this project. I would also like to thank Dr. John Hollywell, Dr. Christopher Minson, as well as all of the graduate students working in the lab for answering all of my questions and guiding me through the process. I would also like to thank the Peter O'Day Fellowship for providing funding for the project.